Thank you for attending and welcome to the second annual Gene and Cell Engineering Summit presented by GenScript. This summit will feature in-depth discussions and research presentations on the crucial gene synthesis and cell editing technologies that are transforming scientific and biomedical research today. Before we start, we would like to provide some housekeeping items. All talks will be available on demand following the summit in the LabRoots auditorium for the next 12 months. Use the chat box to ask questions during the Q&A session after each talk. Exhibit hall and virtual booths are open. You may visit during the break between 1:40 p.m. Eastern Standard Time to 1:55 p.m. Eastern Standard Time and 3:50 p.m. Eastern Standard Time to 4:05 p.m. Eastern Standard Time. Leaderboard will be locked down at 5:20 p.m. Eastern Standard Time, and we will announce the winners at 5:25 p.m. Eastern Standard Time with broadcast messages. For event support or help, please click Help Desk at the bottom of the page. You may also send us a message. Click Career Opportunities for open positions at GenScript to learn more about employment opportunities. Please don't forget to take our customer feedback survey and share on social media using hashtag GCE Summit. Shortly after this introduction, we will welcome Dr. Ray Chen, President of the Life Science Group, who will offer some opening remarks and introduce our keynote speaker, Dr. Guowei Fang, the SVP and Global Head of Research and Early Development at Legend Biotech. Here is an overview of the agenda for today. Following the keynote presentation, we will have talks 2 and 3 between 12:25 p.m. and 1:40 p.m. Eastern Standard Time. This first section will be followed by a 15-minute break. Subsequently, we will have talks 4, 5, and 6 between 1:55 p.m. and 3:50 p.m. Eastern Standard Time, followed by a second 15-minute break. After that, we will have talk 7 between 4:05 p.m. and 4:40 p.m. Eastern Standard Time. Each talk is approximately 20 to 25 minutes, followed by 10 minutes of live Q&A sessions. For our closing keynote presentation, we will have Dr. Pasu Wang, an assistant professor at Stanford University. Lastly, Kei Chuang, GenScript's head of marketing, will present the closing remarks to conclude our summit. Please take a few minutes to become familiar with the lobby. There is a welcome video at the top area of the lobby. There is also a feedback survey that we would like for you to participate in. It can be found in the lobby as well as in your briefcase. There is a chat box in the lower right-hand corner where you can ask questions and also find the help desk for support. And there are also career opportunities that are found in the help desk as well as your briefcase. We would also like you to become familiar with the exhibit hall. There are 5 booths for different services. In each booth, you will find files for supporting resources and ongoing promotions. You may send us a message if you are interested in a particular service, and you may earn points by interacting with our reps. You can claim points by clicking on our 20th anniversary logo that is hiding in different parts of the virtual areas. You can also take a fun photo at the photo booth. Please visit our booths and chat with one of our representatives. Enjoy and have fun. We will tally up the points at 5:20 p.m. Eastern Standard Time, and announce the winners at 5:25 p.m. Eastern Standard Time. The attendee with the most points wins a Meta Oculus Quest 2. We want to take time to thank the GenScript team for making this year's Gene and Cell Engineering Summit possible. And lastly, I'd like to introduce career opportunities here at GenScript. For more information, please find the flyer in the help desk. And now I'd like to welcome Dr. Ray Chen from GenScript.
Hello, everyone. Welcome to our second Gene and Cell Engineering Summit. My name is Ray Chen. We're very thrilled to have you joining us today. On behalf of GenScript Life Science Group, I would like to thank every one of you and our distinguished guests for joining us today. It is unfortunate that we couldn't make this event in person this year, as we would certainly love to. Yet the virtual summit will also connect us to different parts of the world in a very efficient way. This year is GenScript's 20 years anniversary. For the past 2 decades, GenScript has lived a shared purpose. It started as an idea for a better and easy way to make genes and for researchers. Like all other powerful ideas, it evolved into our mission to become the world's leading enabling platform in serving science by providing reliable, high quality, and innovative reagents and instruments. This mission has become what we are passionate about. It is the reason we are on a constant quest to push ourselves to improve and innovate. The impact of what we do goes really beyond business, and the meaningful contributions from us are much larger than ourselves. Globally, there has been thousands of vaccine and therapeutic programs, all started from the reagents we made. We have been supporting almost all the cell and gene therapy research labs around the globe. We are also the pioneers to drive and enable the advancement of long-viral DNA payload for T-cell therapy. In addition, we serve almost all the plant sciences labs and companies globally. More excitingly, with the collaboration with Professor Linfa Wang from Duke and US, we together introduced the first and remain the only FDA-approved test for detecting neutralizing antibody against SARS-CoV-2. That is right. We, GenScript, have built a diverse family of services and product portfolios across a wide variety of existing and emerging life science research and development areas. Each one of our portfolios has its own uniqueness and value that are deeply driven by applications, yet we're bound together by our shared purpose, enabling and helping our customers, the greatest minds in the world, to achieve amazing things. Now, we are more committed than ever to continue developing and building our platform capabilities and capacities around the globe. To better serve our customers, we opened our Innovation Center in Seattle, opened our Singapore facility, and further expanded our US facility that is powered by molecular biology and protein science high-throughput automation platforms. We do hope that we could be more helpful. Once again, thank you so much for joining us today. Today's summit participates highlights the exciting area of gene and cell engineering. We have an agenda that is fully packed with great presentations. Leaders from academia, biotech, and pharma will share with you their latest work developing cancer cell therapies, new platforms for T-cell engineering, and innovative technologies for genome editing. Please take full advantage of this day that have multiple fun activities and also our booth support. Our product managers will be available to assist you with any question you may have. And excitingly, our keynote speaker today, Dr. Guowei Fang, is a Senior Vice President and Global Head of R&D and Early Development at Legend Biotech. Dr. Fang is an accomplished scientist and leader in pharmaceutical R&D. Before his current role at Legend, Dr. Fang was at Zymeworks, where he served as Senior VP of Research and oversaw the development of novel platforms and a portfolio of multi-functional biologics and antibody-drug conjugates. Before that, Dr. Fang led discovery and early development initiatives as head of discovery at Pharmacyclics and AbbVie company. In his current role at Legend, Dr. Fang is in charge of driving multiple exciting pipelines, including solid cancer and direct R&D in United States, Ireland, and China. His presentation today is titled New Innovations in Cell Therapy. So without further ado, welcome Dr. Fang. It is our absolute pleasure to have you with us today. Thank you.
Okay, hello everyone. Welcome to the Gene and Cell Engineering Summit. First, I would like to thank Ray and the organizer for organizing this wonderful summit and for the invitation to present here. The tremendous excitement and progress in the gene therapy and the cell therapy area, and engineering certainly represent one of the key elements that drive all those advancements. What I'm going to talk today is on the cell therapy. Cell therapy is a combined form of gene and cell therapy. The cell therapy has made tremendous progress in the last 6 to 8 years, transformed some of the therapeutic treatment in blood cancer. So if we look at the cell therapy, it's still a relatively nascent therapeutic modality. Small molecule has been around for over 100 years. The first biological therapy was approved about 40 years ago. The first ADC, antibody drug conjugate therapy, was approved in 2002. And ADC used to be regarded as the most complex therapeutic modality, combining both small molecule and a large molecule, until the advancement of cell therapy, which is a live therapy and probably indeed represent the most complex therapeutic modality we have engineered so far. So what does it take to advance and engineer the cell therapy product? It takes a large toolbox. First, it's take tools to generate the antibody and to engineering the right cell signaling module, so-called CAR, as well as some of the element to enhance the immune response, so-called armor. And those elements has to be introduced to the cell, and there's need to be a very robust screening funnel, identify the right molecule. And with all those toolbox, I has to assemble into the right platform, such as we can approach the cell therapy with autologous cell therapy approach or allogeneic cell therapy approach. There are different immune cell lineage, alpha, beta T, gamma, delta T, NK. And engineering is one aspect. Ability to manufacture on large scale at GMP, under the GMP environment is another major element of cell therapy. Another challenge of cell therapy is, to some extent, lack of translation from the preclinical data package to the clinical response, and therefore a direct path to clinical development, quickly generate the proof of concept, as well as leveraging the external key opinion leaders' relationship to advance clinical development is another major element. So what I'm going to do today is really share some of those elements we learned in the past 8 years, and then share some of our advancement in the cell therapy field. So I mean that in the context of Legend Biotech, innovative biotechnology company, as well as the larger learning from the greater field. So at Legend, we have been pioneering cell therapy at global scale. We have global operation in China, in US, as well as in Europe. With all those toolbox I described earlier, we have a compelling pipeline. Our first commercial product was approved early this year, cilta-cel targeting the multiple myeloma patient disease area. We also have a very robust pipeline, preclinical and clinical pipeline focused on hematological as well as solid tumor indications, using two different therapeutic approach, autologous as well as allogeneic cell therapy modality. We are a young company founded in 2014. 2 years later, in 2016, our first cell therapy product entered a clinical trial. A year later, generated remarkable efficacy response with a manageable safety profile, which later becomes cilta-cel. So in 2017, we reached an collaboration agreement with Janssen Biotech. And subsequently, this particular cell therapy product get granted the breakthrough designation by FDA in 2018. And first approval come in early this year, both in US market by FDA as well as in European market through the EMA. So a year of journey, remarkable history, certainly a lot of excitement in terms of what we learned in the cell therapy field. And this is what I want to share with the audience today. In terms of core technology platform, we have the CAR design platform, armor design platform, and we also involve 3 different cell therapy lineage, and alpha, beta, T, NK, and gamma, delta, T. We use both autologous as well as allogeneic cell therapy approach, focusing on blood cancer as well as solid tumor. This slide just illustrates some of the toolbox I mentioned earlier. Every cell therapy starts with understanding the disease, identifying the target. From there, generating the tools, identify the binders through antibody discovery, screening, and then couple the antibody binder with the CAR design as well as armor design, using various in vitro, in vivo functional assays to identify the right cell product. And from there, working with the CMC manufacturer colleagues to produce the cell product, eventually advance into clinic and generate clinical proof of concept data. So the binder itself is quite important. This is one area Legend Biotech does well, and we have a lot of learning coming from past 8 years experience. And I highlight some of those elements here. One is we use a single domain VHH-based binder. And those are relatively small, compact, single chain binders, around 25, 26 kilodalton. And it improves the CAR expression and stability. And it also provides easy access to novel and sometimes hidden epitopes, which are difficult to access by other type of conventional antibody. And it provides robust engineering flexibility because of size, compact size, so that we can design multi-functional, multi-specific CAR. And lastly, through engineering process, we can optimize the immune synapse structure and function, and therefore provide a better immune cell activation. I will illustrate those points with some of the product later in the talk. Manufacture is another key element of cell therapy. At Legend Biotech, we have global manufacturing network, both in US, in EU, as well as in China. All of those collective effort, we have a very robust pipeline focused on the blood cancer as well as solid tumor using both autologous as well as allogeneic approach. And this slide highlights some of our lead program. I will share the recent progress with the BCMA targeted CEDAR cell program, which is highlighted at the top of this slide, as well as some of our growing solid tumor pipeline highlighted in the bottom part of the slides, that including our effort targeting Claudin 18.2 against gastric cancer, targeting GPC3 for liver cancer, as well as other solid tumor indication, and lastly, DLL3 targeting the small cell lung cancer program. So let's focus on our lead product, which is the CEDAR cell. And this is a cell therapy product targeting the multiple myeloma. And this is a disease, quite common disease. About 18% of blood cancer is multiple myeloma. And although there are multiple lines of treatment and different treatment options, there's still very high unmet medical need, as illustrated on the right side of the slide. With all the treatment options available, the median overall survival rate is still less than 12 months for the refractory multiple myeloma patient. And therefore, there is a need for new innovation in treatment in this disease area. And in cell therapy, product research start with identifying the right cell surface target for immune cell to recognize and then to engage. The good news is for multiple myeloma tumor cell, there are a spectrum of cell surface molecule available, and some of them are relatively selective to tumor cell. The one we focus on is called BCMA, and this is a cell surface signaling molecule essential for the survival of long-lived bone marrow plasma cell. And it has a good differential in terms of expression between tumor versus normal cells. It's highly expressed in multiple myeloma tumor cells, and most other cells beyond the plasma cell do not express BCMA. And therefore, it represents a good targeting opportunity for immune cell to engage and to destroy the tumor cell. The cell product we designed is illustrated in this slide, and it's a second generation CAR design. The intracellular signaling domain is 41BB coupled with CD3 zeta. And what's really differentiate this product against the other similar product is the binder itself. First, the binder was derived from the VHH single chain binding antibody, and we also put 2 tandem copy, so 2 copy of VHL, and each copy represent a different binder. The end product is the structurally differentiated the chimeric antigen receptor cell therapy product, as illustrated on the right side of this slide. So how does this product perform? And this slide illustrates one of the registration trial study. And this is called the CARTITUDE-1 study. It is a phase 1B and phase 2 multi-center registration study. We have, this study has been ongoing for multiple years, and the earlier data readout was at the cutoff date at 21.7 months at that time. The overall response rate is 98%, and the stringent CR response rate is at 82.5%. And at that junction, the PFS and OS was not reached. And based on this data, this product called Cell was approved by US FDA early this year for later line, meaning that patients who had been treated with 4 or greater prior treatment, and then progressed. So it was approved for this patient population, and then subsequently, European Commission also approved the same product for patients who have received at least 3 lines of prior treatment. In today's talk, I'm going to share with some of the recent clinical data with the 2-year follow-up. So the follow-up time is extended to 27.7 months instead of 21.7 months at the time of the FDA approval. Certainly, this longer follow-up data continue to support the deep and durable response efficacy profile of this product. So at 28 months of follow-up, you can see that the overall response rate is still at 97.9%, stringent CR rate at 82.5%, very good PR rate, which also cover the stringent CR response patient, is at 94.8% patient. And of 61 patients evaluable for efficacy, 92% of the patients reached the MRD, minimum residual disease, negative state. And this is a very stringent criteria in terms of evaluating the disease state. What it means is that there's less than 1 tumor cell out of 10 to the 5th cell analyzed. And this is a deep response state, so-called MRD negativity state. Most of the patients with high-risk subgroup also respond well. And those high-risk groups, including cytogenetic, based on the cytogenetic profile, high tumor burden, etc. Although the duration of response and the overall survival was shorter among those high-risk disease patient population. This remarkable efficacy was further illustrated in this slide. On the left side of the graph, it represents the progressive free survival rate, and on the right side of the graph represent the overall survival profile. And those data was patient was divided into subgroups. Overall, the median PFS and OS were not reached. At 27.7 months, 54.9% of patients are still progression-free and alive. If we look at the sub-patient population who achieve stringent CR, they have further improvement in PFS compared to overall population, as illustrated in some of those subpopulation diagram. The therapy has a manageable safety profile, as illustrated in this slide. The CRS, cytokine release syndrome, is manageable, mostly at grade 1, grade 2, and 4.1% at grade 3, grade 4, and 1% at grade 5. Similarly, a neurotoxicity profile is also manageable, as shown in this slide. This slide further summarizes some of the safety observations. At the 28th median follow-up time frame, the safety profile was manageable and consistent with the early report of cellular safety profile. There were patients with secondary malignancy, but those patients all have extensive prior treatment, including the immunomodulatory IMiD therapeutics and alkaline agent, as well as autologous stem cell transplant. And all of those prior therapy are associated with increased risk of secondary malignancy. And the other safety observation was the Parkinsonism. And with the improved management strategy, we see a remarkable reduction of the Parkinsonism from 6% down to 0.5% in this longer-term follow-up time. So to just summarize the CARTITUDE-1 study with the follow-up time frame up to 28 months, patients treated with cilta-cel maintained deep and durable response, with median PFS and OS still not yet reached. And the safety profile was consistent with the earlier report and manageable. The overall response rate remained at 98%, with 83% of patients reaching the stringent CR. And currently, the additional clinical development program is shown in the next slide. We have CARTITUDE-2 program, which targeting different patient population to further generate the clinical signal. We have CARTITUDE-4 program targeting the early line of patient, multiple myeloma patients. CARTITUDE-5 clinical trial targeting the transplant ineligible patient, and CARTITUDE-6 clinical program going through a head-to-head comparison of cilta-cel versus the autologous stem cell transplant treatment option. So this is a quick summary of our lead clinical and the commercial asset, cilta-cel. For the rest of this presentation, I'm going to focus on solid tumor. Although cell therapy has made tremendous progress in blood cancer, solid tumor represent another promising area, and yet very difficult to progress disease area. So why is so difficult to make progress in solid tumor settings by cell therapy? There are multiple reasons. One is solid tumor has a very immunosuppressive tumor microenvironment. And second is there are physical barriers which prevent the immune cell infiltrating into the solid tumor itself. And then lastly, the vascular structure and the immune cell permeation into the solid tumor is another barrier. So in order to make progress in the cell therapy for solid tumor patients, one need to address those technical hurdles. Our approach to the solid tumor is summarized in this slide. We start with binder and try to identify the suitable target. And from there, engineer and binder with high affinity and specificity. And this will help us to address the safety and the tox profile in the solid tumor cell therapy space. We know that in the blood cancer, there are various the lineage-specific target, which allow one to target the certain indication of blood cancer. And yet in the solid tumor settings, the lineage-specific tumor antigen are very limited, and therefore we need to focus on tumor selective target, where the expression between tumor versus normal tissue provide sufficient safety margin. And from there began the antibody discovery and engineering work. Second is we need to optimize the design of the CAR construct. And some of the learning we generate from the CEDAR cell certainly is very applicable in this area. Third is we need to enhance the immune response by using or introducing various armor mechanism, which will enhance the immune cell survival, expansion, and persistence. And we would also, those armor mechanisms could also enhance the immune cell infiltration into the solid tumor tissue itself, and therefore overcoming the physical barrier associated with the solid tumor. Those armor mechanisms could also provide the ways to manipulate the immune suppressive tumor microenvironment. To some extent, even convert an immune suppressive signal associated with specific disease into a signaling mechanism which can activate immune cell response, the so-called switch receptor mechanism. And I will illustrate some of those armor mechanisms in the subsequent product I'm going to share with this audience. And then lastly, a component of cell therapy really concerns manufacture feasibility to manufacture, whether it's possible to run at scale, covering sufficient number of patients, as well as the cost of manufacture. So those area is another major focus of continuous innovation. So let's spend a minute on the armor mechanism itself. There are various ways to approach armor mechanism, and we have multiple parallel paths internally. This slide illustrates one of our internal research efforts, focusing on mechanisms that can enhance the T cell fitness, enhance the T cell function, and the so-called intracellular modulator platform. The way we approach this particular platform is through open-ended functional genomic screen using CRISPR library. So we take primary T cell from different donors, introduce a CRISPR library, and then set the stringent functional assay to select for hits which can enhance the T cell fitness. For example, measurement of T cell proliferation and more stringent condition, low glucose, hypoxia condition, also measure the T cell persistence. So out of this genome-wide CRISPR functional screen, we identify a number of different intracellular modulators whose loss of function would enhance the T cell function. One of the hits was illustrated on the right side of this slide. You can see that with loss of function against this particular locus, there is enhancement in mitochondria function. As a consequence of this, there's also a switch in the T cell phenotype, more towards the central memory T cell phenotype. So this is the type of approach we develop internally to identify novel mechanisms which can be applied to cell therapy design to enhance the T cell function and potentially also the therapeutic response. For the next, for remaining part of the talk, I'm going to focus on 3 cell therapy products in the solid tumor indication to illustrate how we take an engineering approach combined with understanding of the immune mechanism, as well as understanding of disease pathology to come up with disease-specific, disease-tailored cell therapy product. So the first product I'm going to share with you is product targeting the Claudin 18.2. And this is a clinical stage product. We term it LB1908. Stranger to this audience, it is a member of tight junction proteins. It's commonly expressed in multiple tumor indications, including gastric cancer and pancreatic cancer. And the expression tends to be at relatively high level in tumor cell. In normal tissue, the only tissue which express Claudin 18.2 is gastric at the low level. And therefore, we develop a cell product targeting Claudin 18.2 using a high affinity single chain binder VHH. And our binder is very specific to Claudin 18.2, does not recognize closely related family member Claudin 18.1. Given the expression of Claudin 18.2 as expression in normal gastric cancer, normal gastric tissue, it's important to fine-tune the cell product design so that we can generate a attractive therapeutic window. In other words, to some extent, differentiate the tumor activity against its activity against the normal gastric tissue. So balancing the safety versus efficacy profile is important consideration in designing this particular asset. Currently, this particular product, LB1908, is in investigator-initiated clinical trial. We already see very promising anti-tumor activity with a manageable safety profile. And this product also cleared the IND filing by US FDA in June of this year. This slide illustrates the overall design principle for LB1908. You can see that it's a single domain VHA binder against Claudin 18.2, very selective. For Claudin 18.2, not recognizing 18.1. And it has a superior efficacy profile in the preclinical study against both gastric cancer model as well as pancreatic tumor model. This slide highlight one of the in vivo pharmacology characterization, looking at the efficacy and the safety profile of the product. The actual model is a gastric tumor model. And you can see that even at the relatively low dose level of 0.3 million cell, we already see very deep response. As we increase the dose, we observe complete remission of the tumor. And this is a durable remission because we do not see observe the regrowth of the tumor. Our binder also recognize the mouse Claudin 18.2, and therefore the pharmacology model also provide insight into the safety profile of our product. We do not see body weight loss, or we do not see major tox signal in this in vivo pharmacology study. Our efficacy profile is also consistent with the in vivo PK exposure. We see a dose-dependent increase of PK, and we see a robust T cell expansion upon infusion of the cell product into animal. As shown, the PK profile is shared here on the right side of the panel. So to just summarize LB1908, currently it's undergoing clinical investigation by the investigator-initiated study. We already dosed up to 3 million cells per kg in human, and encouraging efficacy profile allow us to expand the investigator-initiated study into multiple centers, and the U.S. R&D was cleared in June this year. We're eagerly waiting for additional clinical data for this exciting cell therapy asset. The next product I'm going to share is LB2101. This is a cell product targeting GPC3. GPC3 is oncofetal protein selectively expressing a number of solid tumor indications, including the liver cancer, lung, SCC, as well as germ cell cancers. The design of LB2101 is quite unique. On the binder side, we use a single chain FV, which has very high affinity against the GPC3. We understand that the GPC3 positive tumor have very immune suppressive tumor microenvironment, and therefore we enhance the immune cell function using a unique armor mechanism, which I will share in later slide. And we feel that the inclusion of the armor mechanism is a safe, relatively safe approach here, given that GPC3 is oncofetal protein with minimal normal tissue expression. Currently, this cell product is under investigator-initiated study. We're also planning R&D filing in China for the phase 1 study. The current clinical study is focused on liver cancer, and depending on the clinical signal, we'll decide how to expand into other solid tumor indication. So let's talk about the target itself. Indeed, it's a very attractive target as an oncofetal protein. Its expression is absent in normal vital tissue, and yet it's highly expressed in multiple solid tumor indication, providing potentially providing a treatment option for different solid tumors. On the design principle itself, we use innovative armor mechanism, and this armor mechanism is aimed to overcome the immune suppressive environment observed in the tumor micro.
Hello again. This is Ray from GenScript, and my apology that there might be some technical issues from Dr. Fang's side, and we're trying to solve right now. Hello again. This is Ray. Thank you so much for your patience, and we certainly would love to do this in the future in person, and so that we can be gathered in a room and in the conference places, so that we can enjoy more in-person communications and the presentations as well. Just bear with us a little bit, and thank you so much for your patience.
Hello?
Yeah, welcome back, Dr. Fang. And sorry about the technical issues.
No problem. Yeah. So where do we get the disconnected? Using which side?
I think we were disconnected around the slide 28 or 29-ish.
Okay, so maybe I should start from 28 or 29.
Truly thankful.
Okay, good. No problem. Thank you.
Thank you.
Are we back yet?
Yes, I think so.
Okay. Hello, everyone. Yeah, sorry for the technical issue. And somehow the connection was not stable, and so after redying, it's working well. So maybe we can start from slide 28. So this is a slide introducing our solid tumor asset LB2101. This is a product targeting GPC3. As many of you, GPC3 is an oncofetal protein, and we use a novel armor mechanism to convert the immune suppressive signal observed in the tumor microenvironment into immune activating signal. So next slide quickly show the expression profile of GPC3. And it's oncofetal protein not present in normal vital organ tissues, and yet is highly expressed in multiple solid tumor indications, including liver cancer and non-small cell lung cancer, as well as germ cell cancer. And therefore, it represents a clean target, so-called clean target, attractive for cell therapy. And the main innovation for this product is really the armor design. And we introduce transmembrane signaling mechanism, which can convert immune suppressive signal observed in the tumor microenvironment into an intracellular activating signal to enhance the T-cell expansion persistence, as well as to improve the CAR T-cell infiltration into the tumor tissue itself. This slide illustrates the benefit of introducing armor. This is an in vivo pharmacology study using a liver cancer model. And as you can see on the left panel, there are 4 different cohorts. The black curve represents a control, and then the brownish curve represents unmodified T-cell treated animal. The blue curve is the cohort where we use the T-cell product with the CAR engineered into it, but without the armor mechanism. We began to see CAR-directed tumor killing and observe some anti-tumor efficacy. The last cohort represented by the purple line is so-called H93T. This is a cohort which includes both the CAR as well as the armor mechanism. And you can see the deep, durable tumor remission with H93T. This efficacy profile is consistent with the PK profile. And as you can see that comparing the H93, this is a construct with CAR but without armor, versus the H93T, this is a product with armor. There is a major improvement in terms of T-cell expansion, PK exposure, and to some extent the T-cell persistence as well. The introduction of armor itself does not introduce a safety concern, and it's based on this pharmacology study. And as we measure the body weight, we do not see a change in body weight with the H93T cohort. So just to conclude the LB2101, this is a novel cell product with a unique armor signaling mechanism which convert immune suppressive signal into an immune activating signal to enhance the T-cell function. Currently, this product is under investigator-initiated clinical study for liver cancer, and we are planning to file R&D for the product, and also consider to expand the disease indication beyond the liver cancer. The last product I'm going to share with this audience is a product we call LB2102. This is a cell therapy product targeting DLL3. As you know that DLL3 is promising tumor target, which is highly expressed in small cell lung cancer. And small cell lung cancer itself is a disease indication with a very limited treatment option for targeted therapy, and therefore represent a large unmet medical needs. The binder itself is a VHH-based binder, and to enhance the therapeutic activity, we also introduce another armor mechanism, which is different from the previous product. And this is more of a mechanism to block the immune suppressive effect for the small cell lung cancer. Currently, this product is in the process of preparing for US R&D filing. This slide briefly illustrates the expression profile of DLL3. It's highly and homogeneously expressed in small cell lung cancer, but not in other lung cancer indications, and it's also do not express in normal lung tissue. In fact, it has very limited expression in other normal tissue organs. Binder itself, in a CAR construct, it's a tandem copy of two different binders targeting DLL3, and this is taking the learning from the CEDAR cell to enhance the binding ability using the tandem binder, and also potentially enhance the immune synapse structure and function, and enhance the T cell response. We also include the armor mechanism, which function dominant actively to block the immune suppressive signaling, and therefore provide a mechanism to enhance cell function and T cell response. So what's the benefit of armor? Again, this is illustrated in this pharmacology study. It's a xenograft model for the small cell lung cancer disease. 4 different cohorts, the vehicle and unmodified T cell control arm, as shown in black and gray curve. That's serve as a background control. The blue curve represent a cell product with the CAR, but without the armor. We began to see some disease control. The last cohort, illustrated with the red curve, is a cell product with armor and the CAR construct. Again, we observe deep, durable tumor growth control, and we can eradicate tumor completely for extended time. As we measure the PK profile, the armor mechanism certainly enhance the T cell expansion and persistence. When we look at the histology of tumor itself at the early time point, when tumor are still detected with the LB2102 treated animal, you can see that addition of armor mechanism introduce the T cell infiltration into the tumor microenvironment, as shown in the bottom right panel, whereas a T cell product which contains the CAR construct but without the armor does not introduce much of the T cell infiltration. And therefore, our armor mechanism not only enhance the T cell expansion and persistence, but also has a direct effect on the infiltration of T cell into the tumor microenvironment. Just to summarize LB2102, this is a cell product targeting DLL3, initially for the small cell lung cancer, and the product design involved tandem binder against DLL3 to enhance the immune synapse structure and function. They also involve a unique armor mechanism to block the immune suppressive signaling in solid tumor found in the small cell lung cancer, and therefore enhance the T cell fitness and enhance the T cell infiltration into the tumor microenvironment. Currently, this cell product is in the process to prepare for U.S. R&D filing. So this summary covered wrap up the 3 different solid tumor product. Currently, we are advancing into clinic or are already in the clinical trial and involve unique target selection, involve different armor mechanism to address disease-specific hurdles found in the tumor microenvironment. Just to wrap up this presentation, we think that cell therapy is an exciting area in terms of treatment options for patients. And autologous cell therapy certainly already demonstrated tremendous activities in blood cancer. The next frontier will be in the solid tumor disease indication. We have various design principle coupled with the understanding of immune biology and disease pathology to overcome those technical hurdles in solid tumor indication. We're also deeply engaged or invested in various allogeneic cell therapy platform, which I do not have time to share with this audience today. Overall, I think Legend Biotech is a growing cell therapy company, and we have strong, promising clinical data. We have a global collaboration with big pharmaceutical company with our lead commercial asset. And equally importantly, we have a fully integrated research and development platform internally to drive our next wave of innovation, to continue to move with the frontier of cell therapy, and in certain area continue to lead the cell therapy treatment landscape. Yeah, I will stop here and will be happy to take any questions from the audience.
Thank you so much, Dr. Fang, for your presentation. And Legend certainly achieved quite amazing things for the past years. And congratulations again and to Legend for the Zeta cells approval in Europe and in US. And we do have quite a number of questions coming from the chat box for the Q&A, and we will keep encouraging people to submit the questions. And the first one actually is that the Zeta cell has very impressive efficacy profiles in clinic. And you elaborate a little bit about the elegant design that can drive the differentiated clinical responses. And just wondering, any learnings that from this that can be further applied for the future pipelines, and what we have been trying to make it word duplicate the successes, or that any learnings that we're trying to implement for the future.
Thanks, Ray. Yeah, this is an excellent question, and certainly a question we constantly thinking within our research and development team. And I think a few directions point to the success of Zeta cell. One is really a deep understanding of disease, and starting from selection of the target, and identify a target which has a good expression level in tumor cell, and also a relatively clean profile in normal tissue, so that we could have we can not only achieve good efficacy but also have a clean safety profile. And secondly is the design of the cell product itself. And one of the unique attributes of the CEDAR cell product is the so-called bioparatopic design, where we have tandem copy of binder, two different binders recognizing the same antigen. And those two binders actually recognize different epitope on the tumor target. And this type of bioparatopic design allow the cell product to engage multiple copy of BCMA molecule, and therefore generate enhanced immune synapse. So it's not just the binding event, it's how to translate the binding, the structural element into a functional consequence. And being able to enhance the immune synapse structure, and from there enhance the T-cell response is another key element that the bioparatopic design offered. And indeed, we have structural information, we have functional readout to demonstrate that bioparatopic binder not only improve the binding affinity, drive ability, but has a deep functional consequence. And I think that's probably a second area where CEDAR cell is truly differentiated. And then I think the third area where I think for any cell therapy product, important for cell therapy product is a close collaboration to get clinic validation as soon as possible, and then using that information to feed back into a next round of design to improve your product. And so in general, similar to any other therapeutics, the bench to bedside, and then from bedside back to bench. And this cycle of forward translation and back translation are critical for a successful cell product design and progress. Thank you.
Thank you for the further elaborations about the learnings and truly remarkable achievement for the CEDAR cell, and we anticipate more successes from the commercialization side as well.
Yeah, we are very happy that, thank you. We are very happy that this product is indeed benefiting the patients, and with 98% overall response rate.
Exactly.
Yeah, and above 80% CR rate, and we have patients who are already progression-free for over 5 years. And then to some extent, most of clinicians would regard 5-year progression-free as a functional cure.
Right, truly amazing. Yes. And the second question that coming out from the chat box is that thank you again for introducing about the pipelines and the efforts that the Legend is making for the solid tumors, which represents quite untapped opportunities that, as you said, the global community is trying to tackle. And so far, that the therapeutic responses has been limited, but we're making the journey and to try to get things going. And you elaborated a little bit about the main challenges, including the manufacturing for the cell therapies in solid tumors, and any other particular things or that we're trying, or Legend is trying to do to overcome the technical hurdles, and that you would like to further introduce?
Yeah, that's another excellent question. And solid tumor represent untapped opportunity, and it's an area where there's a tremendous unmet medical needs, and therefore it's patient and physician are constantly looking for new innovations. Looking back on the cell therapy, 8-10 years ago. And the same question was also asked for the blood cancer. At that time, people think that it's impossible to have a live therapy, right? And yet 10 years later, look at where we are, and some remarkable accomplishment for the entire cell therapy field. We think that solid tumor is another opportunity in front of us, and it will have a breakthrough in this area. As a we as a field will have a breakthrough in this area. It's just in what form and shape, when. And what we are really investing in the solid tumor are 3 different areas. One is understand the immune mechanism. The deeper we understand the immune mechanism, the better cell product we can engineer. Second is really understand the disease.
It seems like the internet connection get chopped out once again from Dr. Fang's side, and due to the time limit, that there were quite a number of other questions that people are being asking and through the chat box, and we might not be able to further address today and during the unfortunately for this session, but we will further communicate with the organizer and try to answer those questions offline and from Dr. Fang, and including some further introductions about the intercellular modulators and platform, and also some other discussions as well. My truly apology for the inconveniences, for the technical issues, and well, again, we would certainly enjoy more conversations in person, and we will try to get this event going and in an in-person way for the coming year. But just wanted to say thank you again to Dr. Fang for the presentation, which is truly exciting by laying out the coming pipelines from Legend Biotech, and to really representing a lot of promises that the entire community globally is trying to overcome. So back to the modulator, and let's say that let's move to the next session. We do have a full pack of more exciting updates and presentations from our speakers throughout the day. Thank you so much for joining for the keynote session this morning, and hope you all will enjoy the day.